Hello everyone, and welcome back to the channel. You already know who I am, but today we are going to be doing something a little bit different. We're going to be talking about white nose syndrome. It's going to be educational. Hopefully it will inspire curiosity, but we're going to focus in specifically on the evolutionary history of white nose syndrome and especially one particular quirk that scientists have recently discovered. So it's very exciting, very relevant, and very recent. So to give you an idea of what exactly we're going to be talking about today, we have, first of all, what is parasitic virulence? And so that's the sort of base of this talk, is what is it? And so then after that we're going to move on to meet the pathogen that causes white nose syndrome, Pseudogymnoascus destructans. From there, we're going to look at the scale and effect of this fungus just to see exactly what it does, where it's been doing it, and just how serious and severe it is. Then after that, we'll start looking in at the specific study that this talk is focused on, which looks at a genetic survey of white nose syndrome, the genes it has, what genes it expresses, and then finally we'll look at something particular that this study found that is a potential direction that we can take to stop this pathogen in its tracks. So all very interesting stuff. I love it a lot. Let's jump into it. So first of all, we have to address the question, what is parasitic virulence? This is the whole base of this talk, so it would kind of be good to know what that is. So parasitic virulence, in order to understand it, first we have to understand what a parasite is. So a parasite, to put it plain and simple, is an organism that is in a relationship with another organism where the parasite benefits at the expense of the host. So, in other words, you have two organisms that are in a relationship that interact together. One benefits and the other one is hurt. And specifically there are two requirements that you see here to this. Number one, to be considered a parasite, an organism has to derive its resources from the host that it is currently inhabiting, and the second thing is that it has to reduce the fitness of the host. And so you see this sort of benefit cost relationship here, where this first one, dealing with the derivation of resources, is the benefit to the parasite, but then at the same time you see the host is hurt because it sees a reduction in fitness. And just as a quick review, fitness relates to the ability of an organism to successfully live and survive, to be able to reproduce, and to have viable offspring that will also reproduce. So a great example of a parasite is this nasty creature right here, not the snail, but these worms. And so these worms, the green banded brood or the green striped brood or something to that effect, what they do is they lay eggs in bird poop, or they reproduce in the intestinal tract of a bird. The bird poops, the eggs come out and are in the poop, and snails, as cool as they are, they eat the bird poop, and then they pick up these eggs, which become worms, as you see here. So you'll notice this banding, these look like caterpillars, but these are the eye stalks of the snail in which the worm has fully inhabited and is taking over. What it does is it looks like a sort of flashing disco zombie. Didn't think I'd ever say that string of words. And causes it to go up to tall plants and things where birds can easily see it. It looks like a caterpillar, easy snack for the bird. The bird comes over and eats the eye stalks of the snail. It's very disgusting, but that's a prime example of what parasites do. And so now that we have an understanding of what exactly a parasite is, we can look at the virulence caused by parasites. So, first of all, virulence, just to give a definition, is the ability of a pathogen to cause harm to a host. And so there are a variety of different ways that a pathogen can harm a host. And so some of this variation comes from its competition for resources with the host. So we're looking at what kinds of resources. Most of it deals with nutrition, nutrients, dealing with stealing food sources, food resources from a host, but you can have other such things as well, where, for example, going back to the previous slide, the worms used the snail's body and livelihood. I'm standing under a vent and I don't want the microphone to pick it up. So you can see from the snail, the worm isn't stealing food, it's stealing the snail's ability to live its life without being immediately preyed upon by birds. But then you also have this mechanistic physiological aspect of virulence as well. How exactly is this pathogen, parasite, whatever, invading the host? How is it deriving its resources? Is it burrowing straight into the host? Is it causing disease of some kind? Or is it just simply living its life? One that isn't particularly virulent is known as histoplasmosis. It's a fungus that lives in southeastern US, and people can pick it up, and it only causes pathogenic symptoms in pathological symptoms, in a relatively small number of people. And so then you also have the ectoparasite versus the endoparasite, which is a sort of microcosm within the mechanistic roles of pathogens here. So an ectoparasite is something that lives on the outside, whereas an endoparasite is something that causes a little bit more damage. It's more virulent, so to speak, generally speaking, where it lives on the inside of the host. For example, the worms from the last slide. How are we doing on time? We're doing good. So now we're going to actually look at the pathogen and what it is, just to get an idea. So, like I mentioned earlier, its scientific name is Pseudogymnoascus destructans, and it's often abbreviated as PD. And you can see a photo of how it looks here. Now, this is a relatively new fungus as far as our understanding of it goes. Scientists are really trying to understand it because it is such a menace to the bat population, and we'll get to that in just a second. But because it's so recent, we see that just in 2013, less than a decade ago as of the recording of this video, it was referred to as Geomyces destructans rather than Pseudogymnoascus. So to move forward from that, we see that PD is a sacrophilic fungus, which means it's cold-loving. It only grows in temperatures that range from 4 to 20 degrees Celsius, which for those of you who use Fahrenheit, that is approximately 39 degrees Fahrenheit to 68 degrees Fahrenheit. And that's important for a reason that we'll get into very shortly. Now, evolutionarily speaking, this is a relatively old fungus. It first diverged back in what is now present-day Europe about 23.5 million years ago. And from there, it became a bat specialist. So it only parasitizes bats, and we see that it boasts a nasty 70 to 100% mortality rate in bat colonies, especially in North America. Now, the whole reason that this is an issue is because the fungus first arrived in North America in only 2006. That's 16 years ago. So, just to give you an idea, this is what the fungus looks like once it inhabits the bat. So it colonizes the nose and gives that characteristic look, but then it also, you can perhaps see here, there are different white spots on the bat's wing. Those aren't supposed to be there. That's the fungus. And just to go a little bit deeper into the weeds of the taxonomy behind the fungus, for those of you who are interested, it's part of the phylum Ascomycota, and it's an asexual ascomycete. And then it is part of the family Pseudotiaceae, and this is a not very well studied group, which we'll get into, later, but it also sort of characterizes all of what we know of white nose syndrome. And so, actually, this family includes both Geomyces and Pseudogymnoascus, among other groups, other genera. Now that we've figured that out, let's look at the scale and effects of this disease. What exactly is it capable of? How is it doing it? And where is it going? So, To give you an idea, like I mentioned earlier, this first arrived in the United States in 2006 in the New York State area. And from there it radiated outward, and in just 4 years it had colonized 7 different species. And 2 years after that, just 6 years after the fungus had arrived in the United States, U.S. Fish and Wildlife Service estimated that it had killed a whopping 6.7 million bats in that time frame, which was just astounding. But currently today, we see that there are 12 species that have been confirmed to have the disease in North America, including 2 endangered species, the Indiana bat and I want to say the gray bat, and 1 threatened species. There are 14 species of bats in Eurasia that are also dealing with this disease, and 15 total species worldwide that have tested positive for the fungus but don't show any symptoms of the disease. And so the pathology, I'll show you this photo, or draw your attention to this photo really quickly. You can see that there's a bit of fraying and it looks ashy and almost burned. This is what the fungus does. So it only attacks hibernating bats because, if you recall, this is a psychrophilic fungus, and so it only grows in temperatures of up to 68 degrees Fahrenheit, 20 degrees Celsius. So in other words, bats that hibernate, their body temperature lowers to a low enough point that the fungus can actually take hold and start to infect. So what it causes them to do is it causes them to wake up more frequently as they hibernate, and it causes them to burn through their energy reserves, their fat stores. All the bats that live in at least the United States and Canada are insectivores, and so they eat bugs, and during the winter there are no bugs to be found. So they wake up from their hibernation in the middle of winter, they fly around a little bit, come back in, and their energy stores that much less. And in fact, it's been quantified that they burn through their energy reserves twice as fast as healthy bats. So what the fungus actually does is it colonizes exposed skin, so it can't get through the fur, but it does attack their muzzle, ears, and wings, which gives us the characteristic white nose of white nose syndrome. And from there it breaks down collagen, which is what you can see here. The collagen of the skin is actually being degraded, and it causes scarring that often can't be healed. It's very, very nasty. But with that having been said, hopefully now you see that this is a very, very dangerous fungus for bats to pick up. And so we're going to look a little bit more deeply at the genetic survey of what this fungus is all about. And so we're going to look at the study, but before we do, it's important to establish some base evolutionary concepts that will help us to understand a little bit more about this. So there are 3 here that I want to talk about. First of all, Occam's razor is not necessarily an evolutionary concept, but we see it a lot in the field. So Occam's razor, if you're not familiar, is the concept that you're going to take the path of least resistance, the explanation that is the least complicated, that is the most straightforward, the simplest, that is the one that is most likely going to be the case. You know, there's no need to add a bunch of unnecessary details and things to complicate things, because oftentimes, according to Occam's razor, you're not going to need them. And so the second important concept that we need to cover are trade-offs, evolutionary trade-offs. It's the idea that you can't have it all. You can't have your cake and eat it too. And so what we're looking at is the fact that you and I, every living thing out there, has a certain limited number of resources that we have access to. And so we only have a limited number of things that we can focus on of ourselves to push for example, dexterity, standing upright, having the ability to think better, having bigger brains, having a more complicated digestion system in cows to process more complex food sources, the ability to reproduce quickly and effectively for pathogenicity, that sort of thing. But you can't have it all. That's why you don't see cows with IQs bigger than Einstein or bigger than most humans, because they allocate their resources to developing a very complex digestive tract so they can eat the grass of the fields and actually be able to process it. And then finally, we have this idea of using it or losing it. And so with this, it carries the idea that if you don't use a specific gene or the activity of a specific gene, eventually you're going to not have access to that gene anymore. It's going to switch off and or mutate out, and you won't have access to it. And it sort of ties in with this evolutionary trade-off with respect to limited resource allocation, because if I have two things that, or if, say that I have two genes, right, and one I'm using a lot, the other one I'm not using at all, why am I going to dedicate the resources and energy to producing proteins with this gene if I'm not going to be using it, when I could be devoting those resources to the proteins made with this other gene? It's that idea. So now looking at the study itself, it was done by Palmer et al. In 2018, and what they did is they looked at the genes that PD, Pseudogymnoascus destructans, actually has and expressed, and they characterized them and organized them based on what they did, their functions. And then from there, they also looked at the gene expressions of other Pseudogymnoascus species. Specifically, they looked at 6 non-pathogenic species that derive their nutrients from plants. So what they actually found with this study is quite remarkable. So they found that of the Pseudogymnoascus genus, PD is actually the estranged child, because they found that compared to the other species that they worked with and tested and studied, PD didn't really have very many cazymes, as they're called, which are carbohydrate activating enzymes. Basically what these do is they break down carbohydrates, because fungi don't have mouths, they don't have the ability to eat food like we do, and so they have to find another way to break down nutrients to take them in. That's one way, and all 6 of the other species have it, or express them. PD doesn't. Likewise, they saw that the secreted enzymes of PD were significantly lower by a long shot compared to the other 6. And so secreted enzymes, which make up the secretome, as you see here, basically is the idea, again, that funguses can't eat and can't really take things in, and so they have to break things down and digest things externally before they actually take in the nutrients. But the thing that we're going to be focusing on the most is this right here. They found that PD was lacking in a certain DNA repair mechanism known as alternate excision repair. And so it's very odd what effects that has, but all in all, there was not a single gene family that was increased whatsoever. There were no genes that were made more active, nothing in PD compared to the others. Normally you see some if there's a decrease, but the only thing we saw an increase in, was just repeated sections of DNA, which, as we understand now, is just genetic noise. We don't understand what that does, because there's not seemingly a purpose. And so we're going to get into that DNA repair mechanism with UV light. Could this be a potential fix to this disease, which is currently rampaging through the US, through all of North America, and is killing off bats by the millions? So, you're familiar with UV light. UV light is dangerous. You go outside for extended periods of time and you're told wear sunscreen, because the sun produces UV radiation and you don't want to be exposed to that for long. So, why is that exactly? Well, because UV light, the UV radiation, acts as a mutagen. It causes mutations in DNA. So specifically, you see here with the DNA double helix structure, right? Well, what can happen is, rather than having this structure where you have 2 nucleotides that are bound to each other across the double strand, the ladder, so to speak, instead those bonds are broken and you see a bond formed between 2 nucleotides on the same side of the double helix. And that produces a sort of bump or kink in the DNA structure. As you can see here, this it goes out a little bit, and that causes a lot of issues for the DNA replication and transcription machinery. All that to say, if you can't replicate DNA, the cell is basically broken entirely. So moving from that, we have 3 different kinds or levels of UV light, UVA, B, and C, and those are increasing in power, so to speak, power, frequency, that sort of thing, to where A is the weakest, C is the strongest. Now, with respect to the damage induced by UV radiation comes from the sun. And so in order for life to survive outside of the water at the very least, there had to be some means of repairing the damage done. And so you actually see a variety of different ways, including base excision repair, nucleotide excision repair, the one that we'll be focusing on, alternate excision repair, and photoreactivation, among others. And they each differ in what exactly their mechanism is, but what we see in PD is that there is no alternate excision repair pathway whatsoever. Of all of the different genes that the researchers looked at, PD had no means of AER. And specifically they looked, this gene here, UVE1, was completely missing, where it was present in all 6 other species of pseudogymnoascus. And so we can see here with these diagrams and photos just what effect it has. So the top row is PD, and they just measured different strains of it, and then these 6 here are the other pseudogymnoascus species. And as you can see, as you move over to this photo right here, this is the one that was irradiated with UV light. And as you can see, the 6 pseudogymnoascus species that were non-pathogenic they were harmed a little bit, but not too badly. But there's virtually no PD anywhere to be seen on that final photo up there. And then as you can see with this diagram here, UVA light doesn't really have an effect because they're all pretty prevalent. But then this red line, this is PD. As you can see, as the intensity or the dosage increases, there's a significant drop in the ability of PD to survive. And then you can see it even more so over here with UVC, the strongest form of UV light. There's basically none. By the time you get to this incredibly small dose of 10 millijoules per centimeter squared, there's a 1% survival rate. But the fascinating thing is that PD has other repair mechanisms. It has photolyase repair pathways with the photoreactivation that I mentioned on the last slide, and it also has different kinds of NER and BER. They just aren't active. But for some reason, they're not enough to outweigh the damage done by the UV radiation. And so, to tie into evolution, I'm running out of time here, so I'm going to try and move pretty quickly. I want you to think about this. Why might the AER pathway get lost? So if you want to take a second to think about this, pause the video now. But to answer that, we don't know a specific reason why. However, we do know that, tying back to those evolutionary principles of before, if you don't use it, then you lose it. At least that's what we think. That's what we thought originally, because PD spends its whole life in caves, or most of its life in caves, and so it's not really exposed to UV light. And so you got this whole darkness, no light exposure, and so if it's constantly in darkness, why would it need to repair UV light? But that's not exactly the case here. That's not exactly at play, because many cave-dwelling organisms still have repair pathways for UV light. And in fact, some of them have quite a few. So to sum it up with Pseudotiaceae, the family that I mentioned back on slide 8, we just don't know. We don't have enough of an understanding of this fungus, of the family of fungi, to really have an answer here. But the important thing is the fact that this is research as it's happening. We are looking at these things in real time, and it is essentially a race against time to undo the damage, prevent the damage caused by the fungus in bat populations. And to better understand it, we have to look at the past, understanding why it would have lost this gene in the first place, and its extant relatives, the relatives that it has today, to really understand that. So, really briefly, some future directions that we are looking at with respect to PD and understanding white nose syndrome. We could be looking at potential divergence between the European and North American variants of the fungus to see if there's any sort of evolution happening as the fungus adapts to North American climates, North American species, North American habitats, that sort of thing. Obviously, trying to understand the UV mechanisms, or the UV repair mechanisms, and figuring out why PD doesn't have AER. And then also looking at ways of assessing the safety of UVC or UVB exposure to bats as a means of curing them from the fungus. And in addition, just finding other ways to stem the spread of PD and to make sure that it won't damage the bats as badly, at the very least. But with that, these are my sources. Thank you so much for watching, and I will see you all at some point soon. Thanks.